Leonardo Guasti, so it's your turn to deliver your talk. And Leonardo will talk about reprogramming strategy to generate functional adrenocortical cells. Thank you. The floor is yours.
Yes, I guess you can see my slides. And thank you for the introduction, and good afternoon, everyone. I really appreciate the opportunity to talk and present our data, which was made possible also through funding from IFCA, and also to present a hint of more recent unpublished data. So now, since the kind of original description of adrenal insufficiency by Thomas Addison in 1855 at King's College in London, there's been a sort of exponential growth in the understanding on how adrenal insufficiency could be treated with replacement therapy, as you can see here in this timeline where the main kind of milestones and discoveries are reported. And in parallel, and much more recently, we have witnessed kind of an increased understanding on the biology of the adrenal gland, and this has made possible thinking about alternative treatment modalities such as cell-based therapy or gene therapy, as Stefan was mentioning. So, of course, Stefan has just presented this data a little bit, or in general, and we know that for gene therapy, we need to deliver a functional version of a defective enzyme using, for example, viruses as delivery vectors. And we also know that the strategy works, albeit maybe a bit transiently, in animal models of congenital adrenal hyperplasia, for example, 21-hydroxylase deficiency. And knowing all the kind of recent knowledge regarding adrenocortical biology, especially adrenocortical self-renewal and turnover, we know now how it would be possible to make this treatment a bit more permanent, and that is the need of targeting adrenocortical stem cells. Another strategy which is untested and still under the kind of umbrella of gene therapy would be the delivery of genome editing tools in the adrenal gland. And this is, as I said, an untested strategy. And again, like gene therapy, traditional gene therapy, in order to make this treatment permanent, we will need to target adrenocortical stem cells. So they would generate over time cells that have been kind of cured from the genetic defects. Now, we have been interested in cell-based therapy for quite some time, and of course, in this case, the aim is to transplant functional adrenal cells, and these cells, as Stefan was mentioning, could be xenotransplantation, for example, or they can be the result of reprogramming, so changing the phenotype or using protocols that would allow us to obtain cells with functional adrenocortical phenotype. And of course, this is work in progress, funded also by IFCA, in order to generate these cells using different sources, different starting point, different cell substrate as a starting point, and different reprogramming strategies. Now, we've been focusing mostly in the lineage conversion or transdifferentiation of somatic cells, healthy or from patients with congenital adrenal hyperplasia, transdifferentiation to adrenocortical cells. However, we must not forget that other strategies are possible. For example, the generation of a so-called inducible pluripotent stem cells, which are very undifferentiated cells, which are similar but not identical to embryonic stem cells. And these cells, in theory, using a stepwise approach, could be made to differentiate into adrenal cells. Now, while the slides that you're going to see next are on this part of the transdifferentiation, the kind of generation of adrenal cells via a stepwise differentiation of iPSCs or embryonic stem cells is something that Gerard Ruiz-Babot in Harvard at the moment, later on in Dresden, is working. But I just want to show you, to tell you that somatic cells could also be cells obtained from a patient with congenital adrenal hyperplasia, and again with a step of genome editing, the genetic defect could be cured prior to reprogramming. Now, as I was saying earlier, we have been using different cell substrate for reprogramming. As you can see on the left, you can use skin-derived cells or skin fibroblasts, which are still nowadays the most widely accepted cell substrate for doing these reprogramming experiments, and these are very easy to maintain and to obtain, but you need a skin biopsy, which at the time of this study was not, we were not allowed to do that. It wasn't in our ethical approval, and we couldn't get a skin from patients which were younger than 16 years old. We have also been using blood, and within blood we have at least 2 or 3 different cell populations which have been used successfully for reprogramming to inducible pluripotent stem cells or as a substrate for transdifferentiation. For example, you can use the CD34 hematopoietic stem cells which are circulating, and we have been using the so-called late outgrow endothelial progenitor cells. But mostly we have been using cells which were established from urine, and the procedure is shown on the left. So basically, when you collect urine, you would see, and you plate them, you will see that the vast majority of cells within urine are fully differentiated squamous cells. But if you are patient enough to wait 3 to 10 days and using a very specific media formulation, you will see that some colonies appear within the culture dish. And you have the so-called type 1 colony, which have defined edges, and they do not grow after a single passage. And you also have type 2 colonies, also named urine-derived stem cells, which are characterized by a kind of quite typical rice grain morphology, which were viable for several passages and were bankable. And these cells, as you can see on the right-hand side, were as kind of a classical mesenchymal cell expression of markers, as well as pluripotency markers, but were not positive for any of the hematopoietic stem cell markers, as you can see at the bottom. So these urine-derived stem cells seem to originate from the kidney, because, for example, there have been some studies, if you collect urine from a female which has received a kidney transplant from a male donor, these urine-derived stem cells will have the Y chromosome, and also they display kind of transcriptomic characteristics of the upper urinary tract. So for most of the studies in our laboratory, we've been using urine-derived stem cells, and for a minority of cases, we've been using fibroblasts and blood-derived cells. So, and our was a very much trial and error approach based on what was already present in the literature. This is the protocol that we have been using in order to generate the so-called inducible steroidogenic cells. So we use a lentivirus to deliver quite high amount of a gene encoding for steroidogenic factor 1, which is a very key transcription factor involved in adrenogonadal development and adrenal function throughout life, in the presence of high level of cyclic AMP, which is particularly important for the functioning of glucocorticoid secreting cells, and in order to increase the kind of steroidogenic fitness and cell viability. We also use LHRH and also these other components that you can see here at the bottom. And without going through all the data, but cells which were reprogrammed using this strategy were kind of de novo expressing the vast majority of the steroidogenic enzymes at the mRNA and protein level, as you can see here. And we also use mass spectrometry to analyze kind of a panel of steroids. Unfortunately, at that time, aldosterone was not present. Now it is present with our collaborator at Viapath at King's College. But as you can see here, the same in reprogrammed cells, we could see secretion, de novo secretion mostly of a variety of steroid metabolites. These cells also de novo express the ACTH receptor, melanocortin receptor type 2, together with the associated protein MRAP, and they were responsive to both physiological stimuli such as ACTH, as you can see here in panel C, and pharmacological stimuli such as bromocyclic AMP in terms of cortisol secretion. Now, we've heard already quite a lot regarding the pathogenesis of congenital adrenal hyperplasia, which is a group of autosomal recessive disorders, and the most affected gene is 21 hydroxylases, and most of these CYP21 patients are compound heterozygous, meaning that they have different pathogenic variants on each allele rather than being homozygous, and the parents are normally asymptomatic heterozygous. And here we can see the kind of the steroidogenic pathway, and we know that if you have 21 hydroxylase deficiency, you will have impaired cortisol and aldosterone secretion concomitantly with an accumulation of steroid precursor, which leads, as we know, to increased androgen production utilizing the so-called 5-delta pathway and the CYP17A1. And it is the lowest cortisol that results in a kind of negative feedback inhibition within the hypothalamic pituitary adrenal axis, resulting in a compensatory increase of CRH and especially CA and especially ACTH, leading to hypertrophy of the gland. So these are other examples of genes which are causing CAH. So coming back to our reprogramming experiments, we basically evaluated the steroid profile of patients with CAH, starting from their urine, which we were collecting from outpatient visits in 3 London hospitals. And basically we were performing reprogramming in parallel with reprogramming from healthy donors. And as you can see here, this is patient 1, which has a kind of one of the most common mutation resulting in CAH, which is PI172N, resulting in a simple virilizing form of CAH with a residual enzymatic activity between 1 and 10%. And as you can see here, as expected, you have an accumulation of those metabolites which are prior to the 21-hydroxylase activity, such as 17-hydroxyprogesterone or 17-hydroxypregnenolone, and downregulation of those downstreams, such as cortisol. So having these cells which replicated quite well the kind of phenotypic characteristic in terms of metabolomics of these patients, we tested whether we could restore normal steroidogenesis as normal steroidogenesis, as you can describe it in a kind of in a dish, by delivering the wild-type version of the defective enzyme, again through the use of a lentivirus. And as you can see here, we have more detailed results for patient 1. If you deliver CYP21 enzyme, you have an increase in cortisol secretion, which is kind of multiplicity of infection dependent, concomitantly with a reduction of, for example, 17-hydroxyprogesterone and pregnenolone. And this was also replicated in other patients which had kind of a CH due to mutation in other enzyme. And of course, if you overexpress STAR in a CYP21 patient, nothing happens, and vice versa. Of course, you need to replace the enzyme which is defective. So these cells, which were also kind of viable in vivo upon transplantation, but not using a model of adrenal insufficiency. So these cells can be obtained through a procedure of the lineage conversion. Genetic condition can be modeled quite nicely, and the intervention can be tested in a dish. So now these cells, however, were also all the time mostly proliferation arrested, as you can see here with the PCNA on the right. So we think the way we have done this protocol kind of bypass somehow all the biological events which normally would take place within the capsule and the subcapsular region, where there is a kind of a quite fine interplay between different pathways regulating differentiation of adrenocortical stem progenitor cells towards a proper zona glomerulosa or zona fasciculata phenotype. So this is something that we believe we are missing here, and we could not detect cells which had characteristic of adrenocortical stem cells in our population. The reprogramming is forced, and of course we're using lentiviruses. So at this very moment we are a bit far from clinical application using these very cell types, but it was a step forward. So currently, as you know, this work was done mainly by my previous postdoc, Gerard, who now is in Harvard, and again through funding from IFCA, is really trying to push this project forward, not only by trying to devise a protocol in order to differentiate in a stepwise and more physiologically relevant approach embryonic stem cells and inducible pluripotent stem cells to an adrenocortical phenotype, but also to testing these cells in animal models of adrenal insufficiency. And if I have few minutes left, maybe I can tell you about what we are doing at the moment, which is still in line with potential new treatment for cell-based treatment for adrenal insufficiency. In general, we're interested in this protein, which is called delta-like homolog 1, is an imprinted gene, is known to be involved in regulating early differentiation events, mainly keep cells. It seems to be keeping cells in a rather undifferentiated state, and this is mostly true for example, for hepatoblast. Is a bona fide marker for isolating hepatoblast from the river. There is some controversy relating the kind of the way it acts on cells, but basically we now know that DLK1 expressing cells, which are normally at the periphery of the gland, these cells are indeed another population of adrenocortical stem progenitor cells, and we have demonstrated this using genetic lineage tracing. So these red cells that you can see at the periphery are cells that within kind of 3 weeks or 5 weeks, depending if you're looking on the left or on the right, have migrated from the capsule of the mouse into the cortex and have become with time SF1 positive cells and therefore steroid producing cells. So the kind of phenotype of these cells is being kind of studied at the moment, and we can also say that these cells are a bit dormant postnatally compared to during embryonic development. So these DLK1 cells are therefore adrenocortical precursor in mice, and given the phenotype that we know in humans, it's likely that this is also the case for human adrenals. And but what is important is the first marker of adrenocortical stem progenitor cells, which is cell surface, and for which we have excellent antibodies. So we can isolate this population from mouse or human adrenal. And of course, what we are doing is a characterization from a transcriptomic point of view, proteomic point of view, but we are also attempting to generate this kind of adrenal organoids, which are this kind of miniaturized organs showing kind of realistic microanatomy and functionality, hopefully. I believe we're still a bit far away. In other fields, organoids have been generated, and they quickly made it to new clinical application from different disorders. So I'm going to skip this one, and just to I think I've lost you.
No, we are there. We are there.
I think my connection went for a second. This is just to thank my past and present members of the laboratory, and obviously patients which took part in the study from various London hospitals, and in vivo experiments which were done in collaboration with Stefan in Dresden, and of course funding bodies past, present, and future for this research. Thank you so much.
Thank you very much, Leonardo. And I'm handing over to Pierre Val and Stefan Bornstein for the discussion and the questions.
Well, thank you. Hi, everybody. Can you hear me?
Very well.
Okay, so I guess I should start with questions in the chat. So first, thank you very much for these very interesting presentations, and we can all see that the field has been moving forward quite dramatically over the last 15 years or so. And thanks to the support from IFCA, and from thanks also to really interesting collaborations that have been established through IFCA, and all the support that the association has been giving to that kind of research. I had a couple of questions on gene therapy, because I know it's not going to be covered by Andreas later on. So maybe Stefan, you can comment on that. I was wondering about the viral strategy. Do you think it will ever be long lasting, or that we will have to continuously give the patients some viruses?
Yeah, I mean, absolutely. It's preclinical at this point, but from the fact that there are very few side effects to be expected, the adrenal tropism of these new vectors that are available, experience from other similar diseases, and the fact that you indeed could repeat the treatment, I think makes the whole thing a viable option. Remember, only 30% of the cells, even less, that are transfected seem to be enough to restore normal circumstances, or hormone production that is more or less coming close to what you need in a physiological situation. So I think there is a chance. When we were doing it 20 years ago, we still had problems with the type of virus. There was the Gelsinger case, you might remember. There was a bit of skepticism, and it was an intraadrenal application, a lot more complicated than what currently is available. And so I think it might be really possible to go into clinical applications at this stage without going into larger animals. It's not really necessary to do these kind of things in pigs or in primates, non-human primates. I think I would see that absolutely timely.
And if I can jump in, Pierre.
Go ahead, Leo.
Yes. I think also the ability for us to be able to isolate this kind of adrenocortical stem progenitor cells would also give us some sort of a tool whereby we can screen rather quickly, like different serotypes of AAV, for example. And we know now that, for example, AAV can also be modified in order to target one cell type rather than the other, because the binding of AAV to target cells is rather complex, and there is not just a simple or single receptor. So I think there is room for improvement even for the current strategy that is now being moved to clinical trials.
Right, and I have a couple of questions from the chat and then other questions I can ask. So the first one, which is really related to that, is do we know if the presented approaches could lead to a permanent restoration of the adrenal function? And I guess this is the entire point. I don't know what's your take on that.
So my take is, no, if only I could remember the authors of a quite recent 2018 paper where they show histologically quite nicely that the uptake of this AAV of two serotypes, I think it was 10 and 7, is quite amazing in the zona fasciculata of CYP21 mice, deficient mice, while if you look at the kind of zona glomerulosa capsule, there seems that the trophism of these two serotypes is rather absent. So, as I said earlier, for a long-term kind of treatment, we would need one way or the other to target these adrenocortical stem cells. We don't know if we need to target just 5% or 95%. This will have to be determined kind of empirically.
Yeah, I do think that targeting stem cells is going to be key to really solving the issue. And there was one question from Christine Rougeau. She asked if this cellular therapy could also be possible for Addison patients somehow, which of course is a major issue as well.
Yeah. So there are a few issues with Addison. First of all, the adrenal structure itself might be a bit disrupted because of the pathology itself. But thankfully, I think those with adrenal insufficiency and with diabetes have something in common, that transplanted cells do not need necessarily to be implanted either in the pancreas or in the adrenal. A place which has a good vascularization, for example, like these new devices, is good enough. So, and the second part of the related to Addison being an autoimmune disease, even if I have Addison, I take my own urine cells or skin cells and I reprogram as efficiently as I can to an adrenocortical phenotype, I would still have Addison. The same patient with type 1 diabetes, if they use their own cells, they will still need some sort of drug treatment or encapsulation device, which is immune isolating. So, for example, Biosite, which is this company in California, which is testing now these embryonic stem cells derived pancreatic beta cells for type 1 diabetes, could also be used for patients with Addison disease, for example. So we are maybe, I don't know, maybe 10 years behind what's been doing in another important field of endocrinology, which is type 1 diabetes, in terms of cell-based treatment. But we can learn from their mistake and maybe make it a bit quicker for those with adrenal insufficiency in terms of cell therapies.
Okay. There was another question on how does the gene therapy for CAH developed by Adrenas, I think it's the company that Stefan mentioned initially, compare, and can it be re-administered after efficacy is reduced, and does it eventually migrate to the cortex as you described? I'm not quite sure I understand the last part of the question, but do you know how the two strategies for gene therapy compare?
As far as I know, there's not much difference in terms of efficacy for what has been tested in mice and this new, for what I've seen from a poster of them. So despite the kind of curative outcome of these trials, I would like to see results which are going beyond the expected adrenocortical turnover that, it's 6 months or 4-6 months in mice. It might be a bit longer or maybe the same in humans. So this is something that, these are questions that have not been answered. So I would like to see whether this transiency of the effect due to gene therapy are also true in humans or not. Hopefully not, but I have this fear that if the biology is similar between human and mice, we might see a loss of efficacy with time.